The production of the desired product in biotechnology is not limited only to growing microorganisms. First, in the bioreactor, microorganisms are provided with controlled and optimized conditions so that maximum growth and product formation can take place. First of all, bioreactor pipes. For this, steam sterilization can be used. Its main purpose is to remove unwanted microorganisms so that our desired microorganism can grow without contamination. Now fill sterile nutrient medium in the bioreactor. This medium contains carbon source, nitrogen source, minerals, and other essential nutrients, and uses for metabolism. Now a small starter culture, which is called inoculum, is introduced into sterile medium. Microorganism is present. After this, agitation starts, due to which cells in the whole medium evenly distribute. Agitator continuous mixing does, from sparger oxygen or sterile air is supplied. Monitoring pH and dissolved oxygen is done. If foam is forming during mixing, then anti-foam agent or foam breaker blade or rotator is used to fix it. Now engineered microorganism is not only growing, but also desired product is producing. Then its expression happens, and recombinant protein or other biological product can be formed. Product can be intracellular and can also be secreted extracellularly in the medium. When the level of the desired product becomes optimum, the culture is connected from the bioreactor. Downstream processing through sterile outlet, transfer into the purification section begins. It is necessary to decide where the product is located. If the product is extracellular, meaning it is secreted in the medium, then by centrifugation and membrane filtration, cells can be separated and product containing supernatant can be obtained. But if the product is intracellular, meaning it is present inside the cells, then first the cells will have to be separated and then cell disruption will have to be done. In cell disruption or lysis, mechanical methods such as high-pressure homogenization break the cells and release the desired product present inside. But this does not make the product immediately pure. We get a crude extract in which the desired product is mixed with impurities. The extract is processed using techniques like chromatography and is converted into a usable form. Now the quality control of the final product, potency or biological activity, and important parameters like contamination are checked. If the product is therapeutic or if there is a vaccine, then before human use, applicable regulatory and clinical evaluation may also be required.